Alright, let's go. Number 10. The Martian Methane Cycles. The Curiosity rover was not designed as a life detector. Its sample analysis at Mars Suite, the SAM instrument, was built to characterize the sediment history of Gale Crater and reconstruct the water chemistry of a lake that had dried up billions of years ago. Sniffing the thin atmosphere for trace gases was a secondary function of the tunable laser spectrometer inside it. Standard planetary chemistry is unambiguous on Mars methane. Solar ultraviolet light and reactive hydroxyl radicals destroy any methane molecule in the Martian atmosphere within roughly 300 to 600 years. Any methane detected today must therefore be replenished from a currently active source. There is no way around this constraint. Starting in 2013 and continuing across the following decade, Curiosity began detecting methane pulses. The pulses were not random. They rose and fell with the Martian seasons. Christopher Webster at NASA JPL, in a June 2018 paper in Science, reported a persistent background methane cycle oscillating between roughly 0.24 and 0.65 parts per billion by volume, with occasional spikes reaching approximately 21 parts per billion. The ESA Trace Gas Orbiter, launched specifically to confirm this, reported an upper limit near 0.05 parts per billion from orbit. The rover and the satellite could not agree on whether the gas existed. Explanations arrived in waves. Serpentinization of olivine deep in the crust, possible, but should be steady, not seasonal. Adsorption and release from cold regolith, modeled, cannot match the amplitude. Cosmic dust delivery, the mass is too small. Photolysis of buried organics should also be steady. Subsurface microbial methanogenesis matches the seasonal pattern beautifully, and is the one explanation nobody wants to commit to on paper. The rover versus orbiter conflict may be resolved by methane trapping in a thin, near-surface boundary layer at night, dispersing before satellites can see it, but that itself is speculative. If the seasonal signal is real, and the source turns out to be biological, something is currently exhaling beneath the surface of the planet next to yours. We have driven a rover across Mars for over 4,000 sols, and we still cannot say whether the gas rising past its wheels is the exhalation of rock or of something alive. Move outward from Mars, and the chemistry becomes stranger, colder, and considerably more organic. Number 9. The Titan Chemistry Factory. The Cassini-Huygens mission arrived at Saturn in July 2004 to study the planet's rings, its magnetosphere, and the geology of its largest moon. Nobody on the science team expected Titan's upper atmosphere to behave as an active chemical reactor. It was supposed to be a curiosity, an orange haze over a frozen world. Standard chemistry says cold moons are inert. At Titan's surface temperature of -179 degrees Celsius, chemistry should be sluggish, structural, boring, frozen. There should be nothing happening. Cassini's ion and neutral mass spectrometer found the opposite. Titan's upper atmosphere is manufacturing organic molecules on an industrial scale. Hunter Waite at Southwest Research Institute, in a May 2007 paper in Science, reported that solar ultraviolet radiation and energetic electrons from Saturn's magnetosphere break apart methane and nitrogen at altitudes near 1,000 kilometers. The fragments recombine into complex nitrogen-bearing organic molecules, collectively called Tholins, with molecular masses climbing above 8,000 atomic mass units before they precipitate downward. Sarah Hurst and colleagues at the University of Arizona, in a 2012 paper in Astrobiology, then showed that laboratory-synthesized Tholins, when hydrolyzed with water, produce all 5 nucleobases of DNA and RNA, adenine, guanine, cytosine, thymine, uracil, along with a spectrum of amino acids. Contamination in the laboratory was suspected first. The result was replicated across independent labs using sealed reactors with argon backgrounds and isotope tracking. Analytical artifacts from mass spectrometry were checked with orthogonal techniques. Terrestrial biological input was structurally impossible in the sealed apparatus. What remained was uncomfortable. The ingredients of life self-assemble in a nitrogen-methane atmosphere with only sunlight and cold as inputs. That is not trivial chemistry. That is a moon assembling the parts list. Every ingredient required for Earth-style biochemistry is falling gently onto Titan's surface right now, and has been falling for approximately 4 billion years. If chemistry can build the parts from air, sunlight, and cold, how many other places in this galaxy has this already quietly happened? Chemistry can build the parts. Biology, so far as we know it, chooses which parts to use and in which direction. Number 8. The handedness of life. In 1848, Louis Pasteur was studying tartaric acid crystals precipitated from wine sediment at a factory near Strasbourg. Under a microscope, he noticed the crystals came in two mirror image forms and separated them by hand, one by one, with tweezers. He was not looking for a biosignature. He was solving an optical rotation problem. Almost every organic molecule involved in life has a mirror twin, a left-handed and a right-handed version, called enantiomers. Non-biological chemistry produces the two forms in a precise 50/50ths ratio. That symmetry is thermodynamically preferred and mathematically expected. All known life on Earth uses left-handed amino acids and right-handed sugars exclusively. There is no mixed chirality organism anywhere in the tree of life. Daniel Glavin and colleagues at NASA Goddard, in work extending across multiple papers in Science and Meteoritics and Planetary Science through 2020 and 2023, analyzed material from the Murchison meteorite, from Antarctic CM chondrites, and eventually from the returned Ryugu asteroid sample. They reported amino acid excesses of the left-handed form reaching up to roughly 15% above the 50/50ths baseline in specific isovaline measurements. Sandra Pizzarello at Arizona State had published earlier findings from Murchison in a 1997 paper in Science. The excess was small, but statistically robust. Weak nuclear force parity violation was proposed as the origin. The predicted molecular excess is on the order of 10 to the -17, far too small. Circularly polarized light from massive stars was proposed, plausible, but not proven to persist through planetary accretion. Random symmetry breaking amplified by autocatalysis is mathematically possible, but requires an unexplained first seed. Terrestrial contamination during meteorite handling, controlled for, still shows the excess. And the deepest question, why left-handed rather than right-handed at all, remains without answer. Every enzyme in your body works because every amino acid in every organism you have ever encountered shares the same handedness. Nobody knows why the coin fell that way. If we find a world where life uses the opposite handedness, is that a second Genesis, or a coin that landed the other way? Handedness is one filter. The broader problem is filtering signal from mimicry itself. Number 7. The false positive problem. In the mid-2010s, as JWST's launch approached, a subfield of astrobiology quietly grew around a question the community had been avoiding. What detections in an exoplanet atmosphere would actually mean life? Not would look like life, mean it. A biosignature is defined as a gas that would be difficult to produce without biology. The classic candidates are oxygen, oxygen with methane, nitrous oxide, and dimethyl sulfide. Each is rare in abiotic contexts on Earth. When modeled carefully across the full parameter space of exoplanets, almost every proposed biosignature has at least one abiotic pathway that mimics it perfectly. Victoria Meadows at the University of Washington, in a 2018 review in Astrobiology, cataloged at least 6 independent abiotic mechanisms that generate atmospheric oxygen without any life whatsoever. Sean Domingo Goldman at NASA Goddard, in a 2014 paper in the Astrophysical Journal, showed that hydrogen escape from a water-rich planet orbiting an M-dwarf star produces atmospheric oxygen at levels indistinguishable from those a photosynthetic biosphere would generate. Sarah Rugheimer at Cornell and Cambridge published parallel modeling extending this to methane. Add methane as a co-biosignature, serpentinization can produce both. Add temporal variation, volcanic outgassing varies too. Add spatial patterns, atmospheres mix and homogenize. Add spectral fingerprints of chlorophyll, only detectable if the organism is on the day side, and covers a huge fraction of the surface. Every additional filter removes some false positives and introduces new ones. There is no clean test remaining anywhere in the framework. If we announce life on another world tomorrow, someone competent will have a paper out within 6 weeks explaining how a rock could have produced the signal. How do you prove something is alive when the universe has spent 13.8 billion years learning to imitate life without needing to be? The false positive problem is not theoretical. It is the reason Venus remains an argument. Number 6. The Venus Phosphine Claim. Jane Greaves at Cardiff University was using the James Clerk Maxwell Telescope in Hawaii in 2017 to study Venus's cloud chemistry as a control observation for a broader biosignature survey she was designing. She was not expecting a detection. She was calibrating her methodology. Phosphine, pH 3, is on Earth produced almost exclusively by anaerobic microbes and industrial processes. On rocky planets, standard atmospheric chemistry models predict undetectably low levels. Its presence in an oxidizing atmosphere like Venus's is, thermodynamically speaking, not supposed to happen. The Cardiff team saw a signal at the 1.123 mm phosphine absorption line, apparently rising from Venus's upper cloud deck. Greaves and colleagues in Nature Astronomy in September 2020 reported phosphine at approximately 20 parts per billion in the Venusian cloud layer. Within months, Ignace Snellen at Leiden Observatory reanalyzed the same data and found the signal disappeared with different spectral baseline corrections. Geronimo Villanueva at NASA Goddard, using SOFIA Airborne Telescope data, reported an upper limit of approximately 0.8 parts per billion. The original Greaves team, in a 2021 reprocessing, lowered their claim to roughly 1 to 5 parts per billion. Instrumental artifact from 12th order polynomial baseline fitting, a genuine risk, still disputed by both sides. Sulfur dioxide line contamination near the phosphine feature, a plausible confounder. Volcanic phosphide delivery from Venus's interior, proposed by MIT's Sarah Seeger, unproven. Photochemical production without life, modeled by multiple groups, produces some phosphine, nowhere near enough to explain even the reduced claim. Every explanation is partial. Every rebuttal is partial. The dispute is not closer to resolution than it was 5 years ago. The most Earth-like planet in the solar system may or may not carry a gas in its clouds that on our own planet is a specific fingerprint of anaerobic biology. Half a decade of arguing has not moved the needle. What does it mean that our best telescopes cannot agree on whether a signal exists, let alone what made it? If the argument over Venus proves anything, it is that we lack a shared definition of proof. Number 5. The confidence of detection scale. In 2018, NASA convened a panel of astrobiologists to answer a question the community had spent decades avoiding. What evidence would actually constitute proof of life beyond Earth? Standard scientific process is well established elsewhere. You report a detection. Others attempt replication. Alternative explanations get systematically eliminated. Consensus emerges over years, sometimes decades. For extraterrestrial life, there was no consensus on what the finish line even looked like. Jim Green, then NASA's chief scientist, and colleagues published a proposal in Nature in October 2021 for a confidence of life detection scale. It runs from level 1 to level 7. Level 1 is a signal consistent with biology. Level 4 requires ruling out non-biological sources. Level 7 requires independent in situ confirmation across multiple techniques on a return sample. Every current claim, Mars methane, Venus phosphine, K218b dimethyl sulfide, Enceladus's plume chemistry, sits somewhere between level 1 and level 3. Nothing sits higher. Extraordinary claims require extraordinary evidence, as a standard, vague, unquantifiable. Bayesian priors on the probability of life, both the numerator and the denominator are unknown quantities. 5 sigma statistical significance thresholds borrowed from particle physics may not translate to spectroscopy of a planet you cannot revisit. Multiple independent biosignatures combined. Each biosignature has abiotic explanations, so combining them multiplies uncertainty in both directions. Return samples from another planet, technologically impossible for exoplanets, and decades away even for Enceladus or Europa. The evidence you would need to say we are not alone is for anywhere beyond Mars, technologically unreachable within your lifetime. If we never reach level 7, how many centuries can a level 3 detection sit unresolved before it becomes the answer by default? With a scale that reaches only to level 3, our best current cases are all still contested. Number 4. The Europa plume signal. The Hubble Space Telescope's imaging spectrograph was pointed at Europa in December 2012 to map the Moon's surface reflectance in the far ultraviolet. Lawrence Roth at the Southwest Research Institute was searching for atmospheric variability, routine work on a routine target. Europa is a small, cold moon covered in ice roughly 15 to 25 kilometers thick. Its exosphere should be so thin as to be effectively vacuum. Standard models predict essentially no observable atmospheric activity outside of steady sputtering from Jupiter's radiation belt. Roth's team saw hydrogen and oxygen emission concentrated over Europa's south polar region, exactly where subsurface plume activity would produce it. Lorenz Roth, in a January 2014 paper in Science, reported plume heights up to approximately 200 km, appearing only when Europa was at the far point of its orbit around Jupiter. William Sparks at the Space Telescope Science Institute confirmed the finding using a different Hubble technique, transit imaging against Jupiter's disk, in a 2016 paper in the Astrophysical Journal. Then in 2019, follow-up observations by the same Roth team found no plumes at all. Xianjia Jie at the University of Michigan, in a May 2018 paper in Nature Astronomy, reanalyzed Galileo magnetometer and plasma data from a 1997 flyby, and found a magnetic and density signature consistent with the spacecraft having flown directly through a plume without noticing. Instrumental noise. 3 independent teams using 3 different methods cannot all be wrong in the same direction. Local frost effects cannot generate a 200-kilometer plume. Impact ejecta. The composition does not match. Real plumes venting from a subsurface ocean matches all the data. But then why do they turn on and off? Why did direct imaging so often fail to catch them? And what timing controls their release? Beneath 15 to 25 kilometers of ice, salt water is sloshing against a rocky seafloor with more liquid volume than every ocean on Earth combined. If Europa Clipper flies through a plume and finds organic chemistry, do we announce anything at all, or wait for the sample return that may never come? Europa's ocean is one of 2 strong candidates. The other has already given up more. Number 3. The Enceladus Phosphorus find. Cassini was ending its mission with a series of deep plunges through Saturn's E-ring, a ring composed almost entirely of ice, ejected from Enceladus's south polar geysers. The spacecraft was gathering final data before its scheduled destruction in September 2017. The team assumed there was little left to discover. Life as we know it requires 6 major bioessential elements: carbon, hydrogen, nitrogen, oxygen, sulfur, and phosphorus. By 2018, Cassini had already confirmed the first 5 in Enceladus's plume material. Phosphorus was missing, and phosphorus is widely considered the rate-limiting element for biology across the observable universe. In 2023, Frank Postberg and colleagues reprocessed Cassini's cosmic dust analyzer data recorded years earlier and never fully worked through, and found sodium phosphates in the ice grains from Enceladus. Frank Postberg at the Free University of Berlin, in a June 2023 paper in Nature, reported phosphorus concentrations in the plume material at levels at least 100 times higher than in Earth's oceans. The team identified the specific compounds, orthophosphates, most plausibly dissolved from mineral apatite in Enceladus's rocky core through interaction with the alkaline subsurface ocean. The result was based on just 9 ice grains. The signal was unambiguous. Contamination from Cassini's own instrumentation, calibration ruled this out. Meteoritic delivery cannot account for the observed concentration. Non-biological weathering alone, the leading explanation for the source, and not a failure in itself, but the concentration achieved is far higher than terrestrial analog processes yield. Every checklist item for habitability, liquid water, carbon chemistry, chemical energy, all 6 essential elements, is now checked for Enceladus, every one. There is an ocean 800 million miles from you, where every ingredient known to be required for life exists in ratios more favorable than in the ocean where life on Earth began. Habitable is not the same as inhabited, but for the first time in history, we have run out of chemical reasons to say another world is not. Every ingredient found on another world tightens the pressure of a single older question. Number 2. The Great Filter Argument. In 1996, Robin Hanson at George Mason University, working primarily on economic models of civilizational risk and information markets, tried to formalize why the sky appears empty of aliens despite the age and size of the observable galaxy. If prebiotic chemistry, amino acids, nucleobases, membrane-forming lipids, phosphates, is common across the universe, and if habitable environments are common, then intelligent life should be common. It is not observed. This is the Fermi paradox restated with chemistry in mind. Somewhere between chemistry and civilization, something almost always fails. Hansen named this the Great Filter. Hansen's original essay was self-published in 1996 and later formalized across multiple papers, including work by Milan Ćirković at the Astronomical Observatory of Belgrade, published in Astrobiology across the 2000s. Chemical evidence gathered in the years since, Titan's Tholins, Enceladus's Phosphorus, Ryogi's chiral amino acids, K218b's ambiguous atmosphere, has pushed the filter later in the sequence. Every discovery that shows prebiotic chemistry happens easily is a discovery that moves the bottleneck closer to us, not further from us. Early filter, abiogenesis, is the hard step, requiring specific rare conditions we do not currently understand. Middle filter, the jump from prokaryotic to complex eukaryotic life, took over 1 billion years even on Earth. Late filter, intelligence, civilization, or long-term technological survival is what fails. Rare Earth hypothesis. Peter Ward and Donald Brownlee argued in their 2000 book that specific rare astrophysical conditions filter out most planets before biology even begins. And the anthropic reply: we are here to ask the question, so of course we passed whatever filter exists, but that says nothing about what still lies ahead. Every rock we find carrying the chemistry of life on it is evidence that the reason we do not see aliens is not that life is hard to start. It is evidence that something we have not yet done is hard to survive. If the filter is behind us, we are alone by accident. If the filter is ahead of us, we are alone by consequence. The filter debate stays theoretical until a specific planet crosses the biosignature threshold. Number 1. The K218b spectrum. Niku Madhusudan at the University of Cambridge had spent a decade characterizing the atmospheres of small exoplanets. K218b, an 8.6 Earth-mass planet in the habitable zone of a red dwarf star roughly 124 light-years away in the constellation Leo, was one of the priority targets for JWST's early exoplanet atmosphere observations. Dimethyl sulfide, DMS, is on Earth produced almost entirely by marine phytoplankton. Its detection in an exoplanet atmosphere would be, in Madhusudan's own written words, one of the strongest single-molecule biosignature candidates available in the current framework. In September 2023, and then more strongly in April 2025, Madhusudan's team announced JWST spectra, showing spectral features consistent with dimethyl sulfide and its cousin dimethyl disulfide in K218b's atmosphere. The initial detection, published in the Astrophysical Journal Letters in September 2023, reported the DMS feature at approximately 2 to 3 sigma statistical confidence. The 2025 follow-up, using JWST's mid-infrared instrument, claimed a signal above 3 sigma at concentrations that would on Earth require an active biosphere. Within weeks, independent reanalyses by teams including Raphael Luque at the University of Chicago, Jake Taylor at Oxford, and work led by Xiangmin Cai at UC Riverside argued the statistical significance dropped below 2 sigma with different data reduction assumptions. Stellar contamination from the red dwarf host, modeled, disputed, overlap with other sulfur-bearing species like ethane or methanethiol. Cai's group showed in follow-up work that these species fit the spectra comparably well. Statistical overfitting from limited spectral resolution, the fundamental limit of transit spectroscopy on this specific planet with current instrumentation. Photochemical production of DMS without any biology. Theoretical work suggests comet-delivered or photochemically generated DMS is possible, if unusual. And the deepest problem, 3 sigma is not 5 sigma, and this planet will not be visited by a probe within any time frame we can plan for. The strongest single-molecule biosignature claim in the history of astronomy is currently a statistical argument between rooms full of specialists staring at the same spectrum. If we never resolve whether the signal is real, does the possibility of life on K218b become part of what humans quietly assume, or part of what they quietly forget? The pattern across all 10 is not that the universe is teasing us. It is that we have finally asked the universe a question our instruments cannot yet cleanly hear the answer to. Every gas, every grain, every wobble in a spectrum on this list sits at the exact edge of what we can measure and what we can explain. The chemistry of life is being written across the solar system and beyond, in ink we can barely read. The uncertainty is not out there. The uncertainty is in us. If you want to see more videos like this, click the video on screen.